Hello everyone, today we will be examining the ideas of our article titled The Mre11 Complex Suppresses Oncogene-Driven Breast Tumorigenesis and Metastasis. To review, the Mre11 complex is involved in DNA repair with roles in double-strand break formation, sensing, and processing. In our article, the DNA damage response, or DDR, which is a network consisting of DNA repair, DNA damage signaling, apoptosis, and cell cycle checkpoint functions, was studied to understand its role in cancer prevention and growth. The authors noted that oncogenic stress initiates the DDR, but the mechanism in which this happens is unknown. Additionally, the DDR functions that prevent tumorigenesis are unclear. Using a mouse model, researchers aim to discover how oncogene activation initiates DDR engagement and the DDR's role in oncogene-driven tumorigenesis and metastasis suppression.
The researchers hypothesized that using mutant mouse strains with deficiencies in the DNA damage response could define the role of the Mre11 complex in the oncogene activation response. 3 key stains used in this research include hemotoxylin and eosin or H&E staining, tunnel staining, and immunohistochemistry or IHC staining. H&E staining shows the microanatomy of tissues by staining nuclear components purple and extracellular components of cells pink. Tunnel staining detects DNA breaks that are formed in the final phase of apoptosis. IHC staining detects specific protein markers, which helps classify and diagnose tumors.
Prior research has shown that expression of the new T oncogene in mice results in early DNA damage response activation, hyperplasia, and tumorigenesis. For clarity, an oncogene is a gene that can cause uncontrolled cell growth. Hyperplasia refers to the enlargement of tissue due to an increase in the reproduction rate of cells, and tumorigenesis refers to the formation of tumors. The specific question researchers were concerned with in this study is what aspects of the DDR are most relevant for oncogene-induced tumor suppression. To answer this question, the researchers injected mice with various DDR deficiency mutations using an RCAS virus system carrying the new T gene and observed their cellular response. Injection of the new T carrying virus into wild-type mice resulted in enlargement of the mammary ducts with epithelial hyperplasia. This mammary hyperplasia was correlated with oncogene expression. All of the mutants tested in this experiment were mutants corresponding to specific DDR deficiencies, and these mutants can be seen listed on the x-axis of the shown graph. Out of all the mutants examined, only 2 of them sparked interest in the researchers, as only 2 of the mutants were positive for all 4 hyperplastic markers being looked for shown in the table. The 2 mutants of interest in this experiment were the Mre11 ATLD1 mutant and the NBS1 delta B mutant. And the Mre11 mutant, the entire mRN complex is destabilized. However, in the NBS1 delta B mutant, the NBS1 protein is destabilized, but Mre11 protein expression is unaffected. Both of these mutants have been shown in previous research to be deficient in DDR signaling, DSB repair, and intra-S, G2, and M cell cycle checkpoints. Both of these mutants displayed clearly distinct oncogene-induced cellular responses, as these 2 genotypes both exhibited excessive oncogene-induced hyperplasia that filled the lumen of mouse mammary ducts, as seen in the H&E stains on the right. Also, out of all the cell lines tested, the Mre11 mutant was determined to have the most significant degree of oncogene-induced proliferative change, as seen in the graph. Overall, these results show us that the Mre11 complex plays a critical role in mediating the early response to oncogene activation by restraining the hyperplastic response in mammary epithelium.
To propose the role of the Mre11 complex in DDR engagement, researchers examined the oncogene-dependent activation of the DDR in wild-type and Mre11 ATLD1 mammary hyperplasias. The authors quantitatively analyzed the intensity of immunofluorescence staining relative to nuclear volume in imaging of the mammary hyperplasias to compare the different mice genotypes. Gamma H2AX is a histone variant that forms foci at DNA damage sites and works as a marker to recruit DNA repair factors. 53BP1 is a tumor suppressor binding protein serving as a mediator of the DDR and promotes NHEJ repair. Researchers observed that there was a significant reduction in both NU2-induced gamma H2AX foci formation and 53BP1 accumulation in Mre11 ATLD1 lesions relative to the wild-type lesions. On the other hand, oncogene-dependent DDR activation was not disturbed in mammary glands with the p53 null mutation, yet the p53 gene plays an important role in cell division and cell death control. Based on these results, it was concluded that the Mre11 complex is necessary for activation of the DDR after expression of NU2. To identify cell cycle status of oncogene-expressing hyperplasias, phosphohistone H3 serine 10, also known as PHH3S10, expression was analyzed using PHH3S10 staining. PHH3S10 is deposited at heterochromatin in the late G2 phase of the cell cycle and is thus a marker for cells in the late G2 mitosis phase. The PHH3S10 staining pattern observed indicated that the PHH3S10 signal represented pericentromeric staining that was associated with late G2 cells rather than mitotic cells. Additionally, there was an unexpected four-fold increase in PHH3S10 staining in wild-type hyperplasias compared to Mre11 mutated hyperplasias, suggesting that there is a cell regulation error in these mutated hyperplasias when oncogenes have been activated. The speculation is that because the Mre11 complex is mutated, cells are not arresting in the late G2 phase, which would be indicated by PHH3S10 staining. These data suggest that oncogene activation contributes to an increase in PHH3S10 staining in wild-type cells, but lacks in mutated hyperplasias due to Mre11 malfunctions.
Here, the researchers looked at which stage the cells expressing NuT were arrested by the Mre11-dependent DDR. They used confocal microscopy, which is a fluorescence imaging technique to visualize defined spots in a sample at specific depths, and DAPI staining, which shows where the DNA is in the cell. They then graphed the fluorescence intensity of the cells stained with two signals, PHH3S10 and H3K9Me3. PHH3S10 suggests that the cells are in the G2 phase, and H3K9Me3 is characteristic of heterochromatin, which is seen in mitotically inactive cells. The peaks of the DAPI stains roughly align with both signals, suggesting that the cells are in the G2 phase and are not mitotically active, meaning that they are in G2 arrest. However, the researchers were concerned that the cells retained the signal and still went through mitosis. So they stained centrioles with PHH3S10, which were denoted as positive, while the centrioles without the signal were negative. The fact that most of the positive PHH3S10 centrioles were in the S/G2 phase further supports that the cells are in G2 arrest.
Here, the researchers are analyzing the phenotypic severity of each of the different types of mice. Only 5% of the RCAS NUT-injected wild-type mammary glands developed tumors in a 6-month time period. The CHIK2 NBS1 and CHIK2 and NBS1 developed tumors at a similar rate as wild-type mice. The data for tumor latency in P53515C shows that there is no statistical difference between the wild-type mouse and the P53515C. On the other hand, 40% of Mre11 ATLD1 mice mammary glands developed early-onset mammary tumors in all of the injected glands. Figure 5b shows that the tumor growth rates are equivalent to Mre11 ATLD1 and wild-type mammary tumors with no significant difference.
So Mre11 mutated animals display varying levels of delayed tumor growth. In order to quantify why, scientists studied P19ARF expression, which is an established oncogene-induced tumor suppressive pathway that delays growth. In wild-type, NUT-expressing and Mre11 mutated mammary gland cells were tested for P19ARF expression, and it was discovered that P19ARF was more active in Mre11 mutated glands than wild-type mammary glands by tenfold. Interestingly, NUT expression levels between the wild-type and Mre11 mutated cells were not responsible for the differences in P19ARF expression, and PF3 expression levels were also relatively the same. So because of this, scientists created various frameshift mutations in the INC4A and TRP53 regions and tested P19ARF expression again in these mutants against the control. And after allowing tumorigenesis to continue, it was concluded from the data that P19ARF was selected against over time as oncogenes are activated. This is given by the waning levels of P19ARF expression in Mre11 mutated wild-type glands and Mre11 mutated glands with frameshift mutations in the INC4A locus.
Metastasis is the development of secondary tumors at a distance away from the cancer's primary site. In this case, NUT-driven Mre11 mutant tumors are metastatic to the lungs. We saw previously that tumor growth rates were equivalent in the Mre11 mutant versus wild type. Nonetheless, 70% of the Mre11 mutant tumors developed, shown histologically, meaning through biopsy or surgery, had confirmed lung metastasis. This was not seen in other genotypes analyzed, with a p-value of 0.026. To clarify the mechanism of the increase in lung metastasis in the Mre11 mutant, an experiment was done using serial bioluminescence where luciferase labeled Mre11 mutant and wild-type tumor cells were injected into mice. The quantification of pulmonary bioluminescence at different time points is shown, where the graph on the right shows the respective bioluminescence images and gross autopsy images, demonstrating lung metastatic outgrowth. This showed similar rates of lung colonization for both genotypes with a p-value of 0.07. This finding revealed that the lung metastasis potential of Mre11 mutant cells is not due to metastatic colonization functions, but rather due to an increased rate of metastatic dissemination from the primary tumor.
And so to conclude this presentation, it was discovered that the Mre11-mediated DNA damage response system restrains mammary hyperplasia by affecting resulting in oncogene-induced G2 arrest. It was also discovered that impairment of the Mre11 complex promotes the progression of mammary hyperplasias into invasive and metastatic breast cancers. And lastly, metastatic tumors are often associated with secondary inactivation of the INK4A ARF locus.
Moving to further research, one question stemming from this study was what is the mechanism for how Mre11 hypermorphism promotes oncogene-induced ARF expression? One theory is that Mre11-dependent ATM activation destabilizes ARF in wild-type cells. However, further research is needed to confirm this theory. A second question is how does ARF loss paired with Mre11 impairment promote the metastatic progression of breast cancer? In this study, researchers observed that every INK4A ARF mutated mammary tumor was also metastatic when the Mre11 complex was impaired. Future studies on this observation could give better insight into the mechanism by which cancers can become metastatic.